Welcome to the Deep Dive. Today we're jumping into something pretty groundbreaking in computational biology, RFdiffusion3, or RFD3. It's a deep learning approach that's really changing the game for de novo protein design. Now, older models, like the original RFdiffusion, they were brilliant, absolutely brilliant for building protein structures, the backbones, and figuring out protein interactions. But they kind of hit a snag when the target wasn't another protein. Yet trying to design, say, a protein pocket to bind really precisely with a small drug molecule or maybe a tricky bit of DNA or RNA, that needed a level of detail they didn't quite have. It was like designing with bigger building blocks when you needed atom-by-atom precision.
Exactly. That lack of atomic resolution was the real bottleneck. The older methods were great at placing the amino acids, the residues, but getting those tiny, super specific side chain interactions right, the ones you need to perfectly hold a small molecule, that was tough. So our mission today really is to unpack how RFD3 gets past those residue level limits. It's now modeling every single atom involved, the protein's atoms, sure, but also any other non-protein bits and pieces. And this level of control over the atomic geometry, it just opens up huge possibilities for engineering really complex functional molecules. Think custom enzymes, high specificity binders, next-gen therapeutics.
And here's the kicker, right? Not only did they achieve this huge jump in resolution, but RFD3 actually performs better on lots of benchmarks. And somehow it uses way less computing power. Like maybe a tenth of the cost of things like RFdiffusion2. Now wait, modeling every atom, that should make costs explode, shouldn't it? How on earth did they keep it so efficient? But again, before we get into the speed magic, let's dig into that fundamental shift, first designing atom by atom.
Right. The core change is moving the whole generative process. Instead of diffusing the center of amino acid residues, it diffuses the positions of individual atoms. So right from the start, the model is working at the resolution needed to handle the backbone, the side chains, and any non-protein atoms all at once. The atom is like the basic unit it thinks in.
Okay, but proteins are messy, right? Like tryptophan has loads of side chain atoms, glycine has basically none. How do you feed a uniform structure into a diffusion model that starts from noise when you don't even know the sequence or how many atoms each residue will have?
Yeah, that's a really critical implementation detail they had to solve. So to make it uniform for the diffusion, RFD3 standardizes every single residue. It assumes each one has 4 backbone atoms and 10 potential side chain atoms, so 14 coordinates per residue, always.
10 side chain atoms, even for something tiny like alanine.
Exactly. For those smaller side chains, they fill the missing space with what they call virtual atoms. These are just coordinates, essentially placeholders, put in a standard spot near where the side chain center would be. This gives the model a consistent 14-atom framework to work with during diffusion. Then, later in the process, based on where the actual heavy atoms end up, the model figures out the likely residue identity. It lets the system handle sequence design implicitly while focusing on the atomic structure explicitly.
That's clever. Virtual atoms as placeholders.
So architecturally, what's under the hood of this atom-centric model?
Well, it uses a transformer-based UNet architecture. If you know AlphaFold3, it's kind of reminiscent of the diffusion module there.
Yeah.
But it's been significantly streamlined, really optimized for design tasks, not just prediction. It's got 3 main parts: a downsampling bit, a sparse transformer module in the middle, and then an upsampling module that refines things and spits out the final coordinates.
And managing all that atomic data must be intense. You mentioned innovations in how it handles information. Yeah, 2 key things. First, sparse attention blocks. This is really important. In these blocks, information only gets passed between atoms and tokens if they're actually close to each other in the noisy structure it's working on. Think about it. Protein function is all about local chemistry, right? The precise interactions nearby. So by making the attention sparse, the network focuses only on neighbors. This helps it learn those local chemical rules much better and stops it from getting distracted by global shapes it doesn't need to worry about yet. It prevents overfitting too.
Makes sense. Focus on what matters locally. And the 7 feature, something about cross-attention.
Yes, exactly. They use a cross-attention mechanism for pooling information up and down. It lets the model swap information between the super detailed atom-level view and a slightly coarser token or residue-level view. So the token-level transformer can see the bigger picture, the overall structure, but it can also zoom in and pull specific critical details from just the relevant atoms within that token when it needs to during the denoising process.
Okay, let's get back to that efficiency puzzle. More detail, more atoms, but 10 times faster. That still sounds counterintuitive. How did they manage that?
Right, because a lot of the heavy lifting in older models, especially something like AlphaFold3, came from this big module called the pairformer. It was busy calculating all the complex geometric relationships between every pair of residues. For design, RFD3's creators realized, you know what? We probably don't need that massive compute-hungry thing, so they drastically cut it down. The pairformer went from, 48 layers in AF3 to just 2 layers in RFD3 just for processing the conditioning info. That's a huge cut.
It really is, and the numbers show it. The final RFD3 network, only about 168 million trainable parameters.
Yeah.
Compare that to AlphaFold3, which is around 350 million, give or take, so less than half. Plus, they deliberately left out some really computationally costly operations, things like the triangle multiplicative updates and triangle attention, which earlier models used a lot for global interactions.
So trimming the fat, focusing on local interactions via that sparse attention. Exactly, and the result is pretty dramatic. For typical protein sizes, RFD3 clocks in about 10 times faster than RFdiffusion2. And speed isn't just nice, it's crucial, right?
Oh.
If you're doing high-throughput screening, trying to design lots of drug candidates or enzymes, that speed makes a massive difference in practice.
Okay, so we have this incredible combination. Atom-level resolution for precision, plus it's super fast. That must completely change how you tell the model what you want it to design. This native all-atom approach makes it much simpler to give it really, really precise instructions, doesn't it? Giving the user fine-grained, atomic control over the final output. This is really where it shifts from just predicting things to actual engineering.
Precisely. We're moving beyond just saying, put a helix here or make this region hydrophobic. Now we can talk about atomic level conditioning. Users can specify the geometry with incredible detail.
Like what kind of detail?
Well, things like setting up precise hydrogen bond networks, specifying exactly which atoms should interact with a ligand, defining how a protein should grip onto a piece of DNA.
Can you give an example of how that precision actually leads to a better design outcome?
Sure, take hydrogen bond specification. You can literally tell the model, I want this atom on the protein to be a hydrogen bond donor and that atom on the ligand to be the acceptor. And this wasn't just theory. They trained the model with this kind of explicit atomic guidance. And for designing small molecule binders, actually conditioning on these H-bond requirements, it boosted the success rate, getting the desired hydrogen bond geometry right from about 26, almost 27%, up to nearly 33%. That 6% jump is significant when you're chasing binding affinity.
Wow, okay. So defining H-bonds. What else?
Other cool conditioning methods include specifying solvent accessibility. They call it RSO. Basically, you can tell the model whether a particular atom on your target ligand needs to be buried deep inside the protein pocket or if it should stay exposed to the water outside. They also added center of mass guidance to control positioning, and importantly, they figured out how to use symmetric noise input to reliably generate these really complex, beautiful symmetric structures, things like C3, D2, C5, C7 symmetries.
Right, but with all these detailed specific instructions, H-bonds here, bury this atom there, maintain symmetry, how do you make sure the model actually listens and doesn't just, ignore the tricky parts and generate a generic protein blob?
Good question. They brought in a technique from the world of image generation called classifier-free guidance, or CFG.
Classifier-free guidance. How does that work here?
Think of it like having a strict supervisor double-checking the model's work at every step. During the denoising process, for each step, the model actually does 2 calculations. One is the normal prediction, conditioned on all your specific instructions. The other is an unconditional prediction, basically what the model would generate without any specific guidance. Then it calculates a weighted average between the guided and unguided prediction. By emphasizing the difference, it strongly pushes the final output towards structures that really satisfy all those detailed conditions you gave it. It significantly boosts adherence to complex atom-level instructions.
Okay, that makes sense. It's like constantly reminding it of the specific goal.
Yeah.
So the theory sounds amazing. The architecture is clever.
Yeah.
How did it actually perform? It was trained on the whole protein data bank, right? All kinds of interactions.
Yep, the whole PDB. Protein, small molecule, protein DNA interfaces, they tested it across the board.
And the results.
Very strong. For protein-binding proteins, RFD3 did better than the original RFdiffusion on 4 out of 5 important therapeutic targets they tested. And maybe more importantly, the designs it came up with were much more diverse structurally. It found an average of 8.2 distinct successful structural families, compared to just 1.4 for the older version. Diversity is gold in drug development.
And I guess for things like small molecules or DNA, which can be floppy, that atom-level approach must really shine.
Absolutely. For small molecule binders, RFD3 has a huge advantage because it can design the protein structure and figure out the likely shape of the bound ligand at the same time.
Instead of needing a fixed ligand shape as input.
Exactly. Older methods usually needed you to provide a rigid 3D structure for the ligand, but many drug molecules are flexible. RFD3 designs the pocket while considering the ligand's flexibility, which is much more realistic. And it's a similar story for DNA binding proteins. Figuring out how DNA bends and twists when a protein grabs it is really hard.
Yeah.
RFD3 learns to predict both the protein structure and the DNA's conformation together, just given the DNA sequence. That's super useful if you want to design, say, gene editing tools.
Okay, protein binders, small molecules, DNA. What about the ultimate challenge? Enzyme design, where every atom placement in the active site counts.
Right, that's the acid test for atomic precision. Because RFD3 thinks in atoms, it simplifies the incredibly difficult job of building a protein scaffold around a tiny, precise catalytic motif, like the classic cis-his-asp triad you see in some enzymes. And it excelled here, too. On a standard benchmark set for atomic motif enzyme design, RFD3 outperformed RFD2 on 37 out of 41 cases. That's, what, 90%?
90%, wow.
Yeah, and what was really impressive was its ability to handle complex setups. It generated much better solutions for designs that needed multiple separate functional bits, they call them residue islands, held together, including one really tough case with 7 distinct residue islands, which they managed using that symmetric noise technique.
7 islands, perfectly arranged. That sounds incredibly complex. So the computer simulations look great, but did this translate to stuff that actually works in a test tube?
It did, thankfully. They went into the lab and validated the designs. For the DNA binders, they successfully made and tested some, finding one with a decent binding affinity, 5.89 micromolar. And maybe even more striking, in the enzyme design realm, they created a brand new cysteine hydrolase. It showed good catalytic efficiency with a kcat-Kmem of 3500 to 57, which actually surpassed earlier computational designs aiming for the same chemical reaction. So, theory to practice, it holds up.
So let's wrap this up. What's the big takeaway for everyone listening? RFD3 isn't just another small step. It feels like a fundamental change, right? Treating every single atom as the core unit and combining that precision with that remarkable 10x speedup, it really unlocks a new level of practical control.
I think that's right. The key really is integrating these super-specific atomic-level instructions, defining H-bonds, solvent exposure, active site geometries directly and naturally within the diffusion model itself. That level of precision, combined with the speed, should massively expand what we can even attempt to design. We can move faster beyond just mimicking nature to engineering completely new functions, therapeutics targeting tricky, flexible molecules, or highly efficient bespoke catalysts for industry.
Okay, final thought for you, our listener. To ponder. If we can now reliably generate incredibly complex functional molecules like enzymes with 7 distinct functional regions scaffolded perfectly using this atom-level diffusion, what currently unimaginable, non-natural biological machines or functions might be next on the design table, what impossible problems are about to become solvable. That's all the time we have for this deep dive.